Alright, so I'm going to walk through some of the finer points of transcription translation, starting out with a general overview and then talking about some of the finer details that the book talks about, which might be helpful for you. So, if we get started here, here's some overview videos. I have the volume off on this. This is entering a cell. I'm going to back this up again. There we go. Entering a cell. This structure here would be the nucleus. You can see the little nuclear pores like little holes. As you enter the nucleus, this computer graphic goes ahead and zooms in there. By the way, this computer graphic is about 6 years old, which already makes it outdated, but it does an okay job. Inside the nucleus, you have chromosomes, which are made of DNA. DNA has information for making a protein called a gene, so that segment is called a gene. So somewhere along here, we're going to search for a gene, and we're going to have to unwind and unzip it. The enzyme that actually unwinds and unzips is the old enzyme helicase. So that's helicase working its way down, and it's going to leave an exposed strand up here. Then a number of different initiators are going to hop on there, and this is going to be RNA polymerase. An RNA polymerase, I'm going to pause it, is grabbing these little bits and pieces of RNA that are floating around inside the nucleus. Keep in mind that RNA is A's, U's, C's, and G's. And it is going to systematically and chemically pair together the correct A with U. If A is on DNA, then it's going to pair together a U. If T is on DNA, then it's going to pair it with a C with G, G with C. Okay, so it's clicking these pieces into place. So you can see it clicking away here, and it continues to click these pieces into place. Then what happens is the DNA starts to rewind back on itself. And the mRNA right here, this growing strand right here, is going to peel out of position there. Okay, so now the mRNA has what it needs. It's going to leave the nucleus. There's some processing stuff that we'll talk about later that gets it out of the nucleus, but basically it's single-stranded, so it can fit through the nuclear pores, and out it goes through the nuclear pores. So there goes the mRNA. Now mRNA is going to travel out into the cytoplasm, outside of the nucleus, and it's going to take this message out to a thing called the ribosome. The ribosome is typically 2 subunits. They're going to look like catcher's mitts in here, so like a baseball mitt like this, and then a second subunit is going to come over the top, a little ducky looking thing. Now what it's going to do is it's going to hold this mRNA into place, and it's specifically going to hold codons in place. Codons are 3 base sequences. So those codons, so here's our mRNA. It's going to hold 3 of them in place at a time. And then this molecule here is called tRNA. On one end of it is an anticodon. So if this was like A, this would be U, and it would chemically pair up with it. And then that anticodon on the opposite end of the tRNA molecule, way down here, this thing, that's going to be an amino acid. And that amino acid is what's going to be called for. Basically, this codon is saying, hey, bring me this. Then the next codon in place calls for the next one. This next anticodon pairs up with a codon, these 3 here, and that tRNA molecule brings another amino acid with it. When the amino acids are side by side, they're going to peptide bond. We've learned about how that happens before, so that nitrogen bridge between the 2 amino acids. They're going to peptide bond, and then that bond is going to be so strong it's going to pull electrons away and break the bond here, and this tRNA is just going to eject out, so it gets kicked out. So boom, there it goes out. Another tRNA anticodon with an amino acid kicks out another one, and the process continues. Overall, we keep adding amino acids in a row, kicking out these tRNAs. They'll get recycled later. We'll talk about that at a different time. And the end result then is going to be a growing chain of amino acids. So here's the chain of amino acids here, and here's our mRNA. The mRNA gets cut apart and reused. The amino acids travel and get folded up later on. A little bit higher tech version of this. I'll back this thing up a little bit. Oops. Sorry. Too far. There we go. Sorry. That's the one I wanted to hit. So there are initiator factors and transcription factors that are used on the DNA molecule. This is about 3 years ago, a better computer graphic, but quite a bit more to it. Other transcription factors arrive, so you'll see these little blue you'll see these little colored pieces come in from out of nowhere, basically. So there's a big complex of them. And what they do is they hop on board the DNA double helix, at that TATA box area. So they're going to be on that initiator sequence, and they look for TAC down the road here. Polymerase is going to come along here, and it's going to get in the middle here. And then right at that point, this factor here is going to help it start. It's called initiator sequence. And what it's going to do, we'll talk about it much later, probably another week or two, but it'll help this whole complex spring open. And polymerase then springs out of there. I'm going to back that up about 3 seconds just so you can see it. There we go. So this is going to fold over. It's going to come in contact with that, and then polymerase is going to scream out of there. Polymerase is going to race down the DNA double helix, and that yellow thing that you see that's coming out the top here is your mRNA. So this is actually more real speed, how fast it actually occurs. So a little bit better estimation of the whole thing. It's not as dopey and clunky as the last view gave. So you can see how quickly this adds mRNA pieces coming in through the front and clicking it together out through the top. I'll fast forward here for sake of time. Once it has what it needs, the polymerase stops transcribing the DNA. So the DNA is done being transcribed, and then your little ribbon of mRNA breaks free. Okay, so they'll show that here in a second. There it goes. Now that little ribbon of mRNA leaves the nucleus, and it's going to go out to a ribosome. And again, same kind of concept. Here's the first subunit of the ribosome, and then the second subunit of the ribosome. And again, it's going to feed through there 3 at a time. So you see this thing shifting, and if you paid attention, it's 3 at a time. Then these are our tRNA pieces floating around out in the cytoplasm. And our codons that are on the mRNA are going to be complemented by anticodons on the tRNA. These little pink things that they're bringing with them are the amino acids. So these are actually computer graphic 3D models of what this stuff actually looks like. So these tRNA pieces, these little green things out here, are floating along, and they are going to bring with them different shapes of amino acids. They'll show you the different shapes there. There they are. There's a couple of different ones. Okay. So, and then they'll show you the internal workings of the ribosome here in a second. They'll kind of do a cutaway view here. So here we go. And then inside of there, if you watch it quickly, you can see how there's one that pops in and then it pops out. It pops in, pops out, and then you have this growing chain of amino acids coming out the top. So, same concept, just a little bit more high-tech and digitized, but you get your growing chain of amino acids coming out of the top of, there it is, out of the top of the ribosome. Okay, moving on. A little bit more finer detail that the book maybe help you walk through some of it. RNA Pol 2 needs some help. Sorry, need it back up here. RNA Pol 2 needs some help in getting started. It's not as easy as most of these things show. Usually it's promoter region, where that bendy little thing that came down, that usually is the TATA box. It's called TATAAA in succession. It helps polymerase find a starting point. So keep that in mind. Promoter regions, TATA box are important terms here. Also, proteins use these, and they're called transcription factors. So there's transcription factors that look for that TATA box, hop on board, and they form like a conglomerate of things. So here's like 1, 2, 3, and then here's polymerase. So polymerase looks for all these transcription factors hopped on board early on. Continuing on, when RNA is done, when mRNA is done being transcribed, it's not really done. It's kind of a mess. There's a lot of unusable nucleotides. Those are called introns, buried in the usable code. Those are called exons. We think it's leftovers from viral infestation. That just basically gives us junk DNA in the middle of genes. So spliceosomes have been developed over time, and they're enzymes that use small nuclear ribonucleoproteins, affectionately called SNRPs. I don't know how you get SNRPs out of this, by the way, but you do. They use SNRPs to identify intron sequences and loop them. So here's a diagram from the book. This darker red area up here, that is going to be our intron. This is our intron. And here's an exon. This would be a sequence of nucleic acids on the RNA, on the mRNA, that isn't actually going to be used. So they hop on board, these little SNRPs, hop on board and grab onto this at various locations and basically fold it against itself. So they fold it against itself, and then this enzyme back here called a spliceosome, this enzyme then cuts it here and here, and brings that end right here to this end over here. So now you have intron or exon connected to exon. Okay, and then you have your intron out of there. Okay. Final mRNA editing. Before the mRNA leaves, it's given a 5' cap of guanine triphosphate, which is GTP. So GTP. Oops, sorry, that P. The phosphates are used for energy, so it actually helps them open up those gates as it leaves the nucleus. The mRNA then gets also a 3' poly A tail to end up, and you'll see what the importance of that is here shortly, hopefully. Now it's got everything it needs, the tickets are done, and it goes ahead and leaves the nucleus. Okay, so in translation, there's a little bit of details in the ribosomes. The ribosome is made up of ribosomal RNA twisted in a 2 subunit form. There's a large one and a small subunit, so you'll hear the book talking about that repeatedly. Inside, there's a small pocket with locations that they've labeled E, P, and A. A is where it arrives, P is where it's processed, in other words, where the amino acids bind to one another, and then E is where it exits, where the tRNA exits. So you're going to see diagrams that kind of look like this, where there's these 3 pockets, 1, 2, and 3. I don't know what this thing is. Again, different diagram, same idea, 3 pockets. And again, going back to that diagram, this is the one the book uses, I believe. You have the A site where this amino acid is being brought in, or it's arriving. You have the P where it's being processed into the polypeptide chain. So in other words, it's growing an amino acid chain. And then you have the E site where it's exiting or ejecting. Again, codons on the mRNA are calling for specific anticodons, and then those polypeptides are moving along. Okay, and finally the recycling of material. Once tRNA drops off what it needs, it's chemically drawn to another enzyme, aminoacyl tRNA synthetase. It rejoins naked tRNA molecules, ones that have already dropped off their amino acids, with their correct amino acids. So they're amino acid specific. Here's generally the idea behind it. Here's that aminoacyl tRNA synthetase. Here's its active site, right? Here's an amino acid. And what it does is it uses the energy of ATP to basically shove that amino acid into its little spot. It leaves it with 1 phosphate intact. Then tRNA comes along, and using that 1 phosphate, it uses that energy to bond with the amino acid. Then the tRNA is going to leave the complex, and it is now charged. So it's going to go back to the whole ribosome unit and drop off this amino acid. And then once that amino acid is used, then it gets recycled. Everything gets recycled here. Okay. mRNA nucleotides, when they're done, when the mRNA is done out in the ribosomal complex, they get just broken apart, chopped back up. The A's, the U's, the C's, and the G's go back into the nucleus because they're in higher concentration in the cytoplasm, and they go back into the nucleus. All right. So here's an assignment for you. To gain an understanding of where you're at. These are the different levels of understanding as I would predict them to be. Go ahead and take a stab at any one of these levels or all of them. Realize that the 4 level means, yeah, I've left out some stuff here for you to try to figure out and decipher and decode, but you should be able to get to that. So ultimately, that's the goal. I did want to mention about the long chains of the poly A tail. The reason that the poly A tail is important, I'll just do it right now, is because you need a long chain of stuff back here. Even when this is a stop codon, you need something here to hold a place. Otherwise, the ribosome just loses its grip and the whole thing falls out. So if you don't have that poly A tail there, and you just have a stop codon, the thing will just fall through and you'll never get a stop. So that's the reason behind that. Anyway, back to your questions or your practice, and there you have it.